Back in 1987, there were 14 dead humpback whales in Cape Cod Bay, and at the time we knew hardly anything about harmful algal blooms. In fact, the only thing we really knew is that in Japan, Demoic acid was actually used to get rid of parasites in kids, and how they figured that one out, I don't know. And certainly I've seen dead marine mammals full of Demoic acid that are still full of parasites. I don't know, but that's the thing. So in 87, there were these dead whales, and there was a vet called Dr. Joe Gerasi, and he looked at water samples and saw that there was this big algal bloom, and recognized that obviously mackerel could be eating this. So he just hypothesized that perhaps these whales had been poisoned by mackerel that had eaten the plankton that were producing saxitoxin. We have no idea about levels. There was no background information. So what he did is he got the stomach contents of the whale and had them analyzed for saxitoxin, and it was there. So, but there was no way of knowing is that level important or not. So there were no real tests at that time. So what he did is he took some of the whales' some of the whale's kidney and some of the whale's stomach contents, and he fed them to mice, and then the mice died. So he said, well, it's toxic. So then that was 87. So then along come all the clever people in the 90s, and they go, well, of course the mice died. The whale was full of Clostridium, and the whale was full of all these other things. So it probably wasn't the saxitoxin. So that all went away, and 87, the 90s go by, the 2000s go by. Now we come to 2000 and whatever we were, 15 last year. There's more dead whales. So now we go, oh, well, maybe it was the biotoxins. So we start taking samples again and having another look. So you really got to start, it's like all poisons, right? Low doses are not a problem, and then a little bit might be a small amount of problem, and a high dose is a problem. So you've got to start understanding what is the range that we can tolerate too. So that's what we really knew about whales and saxitoxin, was this one event back in 1987.
So are these toxins that only exist in soft tissue?
Actually, these aren't. It depends on the toxin. Some of them aren't even in soft tissue. So Domoic acid, which I'll talk about in a minute, some of them are water soluble and some of them are fat soluble. So the fat soluble ones, you eat them, they're fat soluble, they cross the intestine, they get into the bloodstream, and they go into fatty tissues. So they can go to liver and kidney and blubber. The water soluble ones, like Domoic acid, they can only get out of the intestine by binding to a specific receptor, because it's a fatty membrane. And then once they're in the blood, they're actually excreted through the kidney. They're filtered out really quickly. So Domoic acid, if you give it to a monkey or a rat in the lab, it's cleared from the bloodstream in 4 hours through the kidneys, and it ends up in the urine. And then in the urine, it sits there until you pee it out. So we've found in sea lions that it's not in the soft tissues at all. There's none of it in the soft tissues. It's in the guts and it's in the urine. So the rest of the animal hasn't got any Domoic acid. But it has to have a high enough level in that 4-hour period to bind to some of these glutamate receptors, and that's where the damage occurs. So it varies. So saxitoxin is fat soluble, so it stays in the tissues. Sorry.
Yeah. So damage.
I'm going to get to damage.
Oh, great. Okay.
We'll get to damage. I'm trying to kind of work on with it. So saxitoxin. There we were. Joe Gerasi did this great work, but who knew? And then in 2008, in the St. Lawrence River, just between August the 5th and August the 18th, just over a 2-week period, there were 10 dead beluga, 8 dead harbor porpoise, a dead fin whale, 39 gray seals, 12 harbor seals, lots of aquatic birds, loons, alcids, fulmars, and all these animals, plus invertebrates that were sampled, specifically whelks and crabs, and then fish, they all had high levels of saxitoxin. And so that was really the first sort of new event in recent years where there was this big algal bloom in the St. Lawrence and these dead animals. And nobody really wanted to believe it, because they thought, well, it's always been there and there's got to be other things going on, and we know in the St. Lawrence we have all these contaminants that cause cancer, but there was nothing else. There was no infectious disease found in these animals, and it just lasted for 2 weeks, and then as soon as the bloom went away, it went away. So it was this sort of, it becomes the circumstance. You've got the timing of it, plus all the different species, plus the bloom, plus nothing else, that in the end, you can probably conclude that was probably a big event caused by saxitoxin. But why did it happen just then? It's a confluence of circumstance. Okay, so now we're going to get to effects for domoic acid, and the reason I'm going to focus on domoic acid is because California sea lions are really common along the California coast, and these domoic acid producing blooms have been really common for about 20 years. And so California sea lions will eat anchovies and sardines that contain plankton and domoic acid. But because they're sea lions, when they're sick, they come up on the beach. And then because in California we have a lot of people who care about marine mammals, they see a sea lion on the beach, they telephone, or they get on the internet, and they come into the place that I work. So it's a really good way to study the effects of these algae on the animal, because we can see it on the beach, in the hospital, as it dies, and we can then sample them at different intervals. So we've been able to get a lot of data on this species, but mostly because of its behavior. And in California, we also have whales and we also have dolphins. They're dead, they're floating offshore, and we're still struggling with trying to understand what the effects are, because they're dead, they die at sea. By the time they wash up, their brain's liquid, and we can't understand what's happening. So I'm giving you this as sort of the best information we have on domoic acid, but this is in sea lions, and we really don't know about, can we translate all this to whales? So this was, the first time we really found it was in 1998, and it was in one weekend. So the chlorophyll image is enhanced for red for chlorophyll. So we see there's a big red bloom, and then in that there's these white dots, which are sampling stations where water was collected. And I say we, there are about 20 different organizations involved in this, one working on water, one on the toxin, one on the fish. So those white dots was where the water was collected, and high levels of the plankton were seen. And then plankton was seen, and the anchovies collected by Department of Fish and Game had high levels of domoic acid. And then we had all these piles of sea lions all piled up like this. So these animals were all having seizures and convulsions. And they were doing that because the domoic acid crossed from the bloodstream into the brain and then bound to neurons, cells in the part of the hippocampus that has high numbers of glutamate receptors. So it was only in the bloodstream for a very short time, but in that short time it went to the place with the most glutamate receptors, which was the hippocampus. And hippocampus is involved in seizure activity, so these animals all had convulsions, and most of them died. So domoic acid poisoning was first actually recognized in people in 1986 off Prince Edward Island in Canada, when there was a batch of contaminated mussels that was fed to about 100 people. So of those 106 people, 4 people died, but a couple of the survivors developed epilepsy, and they also had problems with memory. So ever since then, domoic acid poisoning has been called amnesic shellfish poisoning, because survivors of the 1986 event off Prince Edward Island had memory loss. But that led us to wonder, well, I wonder if these surviving sea lions are going to have memory loss. How would we know? So this is what happens in the acute, these animals that were first affected. And so they're pretty severe seizures, but this animal is on land. So you can imagine if that was a dolphin or a whale doing that at sea, it would simply drown. So you wouldn't be able to recognize what was happening. But it's distressing to see, but it's very dramatic. So that's a sea lion having an acute seizure. Then in the animals that died, these are 2 different brains. On the left is a brain from a normal sea lion, and this part of the brain, the hippocampus, it's named that because it's shaped like a seahorse, and hippocampus is the Latin name for seahorse. And on the right is a sea lion that survived a few months after domoic acid poisoning and then died. And what you can see is, instead of that big horseshoe seahorse-shaped structure, there's this tiny little dot that's completely shriveled away. So that sea lion survived the seizures, but the part of the brain, the hippocampus, that has all those glutamate receptors was basically destroyed. Those neurons did their little spasm, not enough to kill the animal, but then they killed themselves. So those neurons are gone. That part of the brain is damaged and is gone. So what I think is a really cool story is in London, we have cab drivers that have to get really fast around London between the theatre and the restaurant. And for them to do that, they have to pass an exam, and it's called the London, it's like a taxi driver, it's called the test. And they can memorize entire maps of London. So if you get into a cab in London and you say, get me to the theatre, they can get you down these back streets and really quick. So a recent study did MRI studies of London taxi drivers' brains, and London taxi drivers have bigger hippocampi than the rest of the British public. So it's developed for spatial navigation and memory, and it can actually expand, and it's very plastic. So that's pretty cool. But it makes us wonder, well, what happens to these animals if they don't have a hippocampus? What are they going to do? So what we did, again, we're lucky because we have animals, near a big city. So we took them to a hospital at night, and we put these sea lions that had recovered through an MRI scanning unit, and we could scan their brains. And on the bottom, on the left, is a normal brain, and then on the right, there's a sea lion that its hippocampus is shriveled, but this sea lion is still alive. This is an MRI, this animal is still alive. So we were trying to find out what would happen to an animal if it didn't have a hippocampus. So we actually took 20 of these sea lions that had survived, we scanned them, checked out their MRI, and then we released them with satellite-linked transmitters in their back to see where they went. And then we could tell, where they went. So on the right is a map from one of those animals. That's the California coast. And instead of staying coastal and going up and down and maybe going to the islands and seeing its pup, this animal went straight west and went towards Hawaii and then disappeared at sea. And so we did this for about 20 animals, and none of them were basically able to. They didn't move in the way that sea lions should. They either went straight west out to sea, a couple of them went up rivers, and it was clear that there was something really abnormal with their spatial navigation, which would fit with losing your hippocampus. So what we think happens is the ones that have a high dose have seizures and die, but the ones that have a low dose, their hippocampus is damaged and their ability to navigate is compromised in the future. And then the other effect that we found is that there are glutamate receptors on the uterus and in the heart. So since 1990, in the first years we were studying this, 1998 to 2000, 274 of the stranded sea lions were pregnant, and then they aborted. And so we looked at the uterus, and sure enough, there were these receptors. But what is more important, that when they aborted, we tested fluids from these aborted fetuses, so the stomach contents or the amniotic fluid, and what we found was domoic acid in these fetuses. But some of these sea lions had been rescued and had been in the hospital for 2 to 3 weeks, and so the domoic acid should have been gone, but we were finding it in the fetus. So what we think is happening there is that the sea lions eating the anchovies, the domoic acid gets into the bloodstream, and then that bloodstream crosses the placenta and goes into the fetal bloodstream. So now you have this fetus that has domoic acid in its bloodstream, but its bloodstream is cleared by its kidneys, so that domoic acid goes into the fetal kidney, which goes into the fetal urine, which is peed out into the amniotic fluid. So once it's in the amniotic fluid, all that can happen is that fetus swallows the amniotic fluid, and the domoic acid just goes around. So since then we've been doing more on this, and we've found that domoic acid can stay in the amniotic fluid for basically the entire gestation. So if an animal eats a little bit of domoic acid in January, it's really not that much, but its fetus is going to be exposed for 6 months until it gives birth in June. So this was one of the concerns that happened this year with the crab fishery in California, was the human health department was worried about these really low levels for pregnant women, because if the same thing happens in people, you could get this very low level that's not that important, but it could accumulate in the amniotic fluid. So that is data that we have from sea lions.